I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of Kulin Nation, and pay my respects to their elders past and present, and the elders of other communities that might be here today. It's a great pleasure to introduce Colby, Professor Colby Zaph, to give our lecture for the postgrad lecture series today. Colby did his PhD at Penn University in Philadelphia and then stayed on to do his postdoc work with David Artis there. He then moved to start his own lab in 2008 at University of British Columbia in Vancouver, and then was recruited to Monash University in Melbourne to continue his work that looks at immune responses of mucosal surfaces and the regulation of methylation. And I've also just been informed that Colby is a ice hockey champion. And so with that really stellar CV, I'd like to hand over to Colby today.
No, thank you for the invitation. Yes, we did win the Australian over 35 championship last weekend. So I was going to post pictures of the trophy and everything, but no. But it's mostly Canadians playing against Canadians, so it doesn't really count. But no, thank you so much for letting me come into the city today through the traffic and the rain. I was talking to Ethan this morning. It's like driving in Vancouver again, just with the rain coming down and everything. So, I'm going to spend some time today talking about some recent data that we've generated, focusing on one aspect of the lab, which is sort of overarches the whole part of the lab, which is this idea of biological methylation. And we say biological methylation because when you start thinking about methylation, the traditional thought is DNA methylation, histone methylation. But it's clear that methylation is really becoming the new phosphorylation. And it's the idea that it's a dynamic post-translational event that can affect all proteins in the cytoplasm and in the nucleus that can really dictate cellular function. So I've got a couple of examples of how biological methylation affects both histone and non-histone proteins today. So we focus on mucosal sites, but primarily the intestinal microenvironment. And I put this slide up there just to remind me to tell people that maybe don't think about the gut as much, is that it's a pretty complex and pretty fascinating place to look at both immunity and immune cells and non-immune cells that I'm going to talk about today. It's this huge interface, which is a single cell layer of epithelial cells that line the gut, that are interacting with what, for all intents and purposes, is the outside world. So this is bacteria, commensal and pathogenic, viruses, fungi, parasites, every sort of pathogen and organism that you can imagine, as well as this interaction with food. And so determining what is pathogenic, what is commensal, what is food, even food that you've never had before is a foreign antigen, yet we don't respond to it as often. And of course, these food allergies are increasing, but in general, most people don't respond to these. And so how are those decisions made? Really, this interplay between the epithelium and this huge amount of immune cells that are found within lymphoid follicles, Peyer's patches, as well as the lamina propria itself, and the interaction between these environments is really sort of what sort of led me to sort of start working on this, or the aspects of this. So in our lab, we sort of focus on there's three general projects that are going on in the lab right now, and I'll talk about a couple of them today. This is the work where methylation affects the histones, and so looking at how methylation of histones impacts on gene expression, and we're going to talk about some of the lymphoid cell development and function in the context of that. And then the first part I'm going to talk about this enzyme here called SET7, which was originally found to be a histone modifier, but really it's clear that it's not. It seems to be doing a lot of other things, and we'll talk about its role in really focusing on non-immune cells and epithelial cells as well. I don't have time to talk today about this project, which is the role of retinoic acid that we have. We've identified a transcription factor downstream of retinoic acid that seems to be really important in controlling immune cell function in the small intestine, but I'm happy to talk about that one if anybody wants to talk about that afterwards. So methylation, when I talk about biological methylation, really I'm talking about the addition of a methyl group to lysines. And this can be N-terminal lysines, this can be embedded lysines. This is at the epsilon amino terminus of this protein. And again, it's just one of a whole range of modifications that can happen to lysines. The interesting thing about methylation of a lysine residue is that this can be mono, di, or trimethylated at this lysine, at this nitrogen, but this never loses its charge. It maintains its charge. So it changes the substrate, the structure of this lysine without changing its charge. Whereas something like acetylation, which is it can be monoacetylated on here, will reduce this charge to, will lose this charge based on the negative charge of that. So it's changing the biophysical properties of a protein without changing its net charge. And so that sort of has ramifications in interactions with other proteins and other things. But really, it suggests that this is really creating a new binding site for a whole range of factors that can bind to methylated lysine residues. So the proteins that mediate this methylation event are called the set domain family, and there's about 45 of them, I guess, at last count. And you can see that there's a whole range of them here. I think I'm losing that. And these are all grouped here by their structural homology, but you can see that even as the structures are grouped together, they are tended to group between their specificities as well. So things like the mixed lineage leukemia family, the MLLs, which are homologous to the Drosophila trithorax protein, they tend to methylate lysine 4 of histone 3. So that's the nomenclature, H3K4. Whereas the EZH1 and 2s, you'll know work from Reese and Stephen, that this tends to be the main focus is the K27 mark. The protein I'm going to spend time on later is this one, G9A, which is H3K9 dimethyltransferase, which is part of the SUV39 family. Also some of this work from Reese's postdoctoral fellowship. And you'll notice this one down here, SET7. This is the one I'm going to spend the first half of the talk on. And you can see that it's very unlike all these other ones, in that it's very early. It's not found in Drosophila. It's not found in yeast. It's only found in higher eukaryotes. And it was first characterized as being a methyltransferase for lysine 4, which I think I'll show you it doesn't happen. But its real claim to fame was that it was found to methylate P53. And a lot of work went into showing that, if you don't have this methylation event, P53 becomes non-functional. And it was quite a good big stir of stories following that. But I think what's clear is that that was not true. So what's going to show you is that we've lots of data. And a lot of data I'm not going to even bother showing you that it doesn't seem to affect that. So here's SET7, the data on SET7. This is the wild types. This is the het. This is the knockout. These are just total knockouts. These mice are absolutely fine. They live normal lives, the total knockouts. We can delete the antibody, but we can see we have really little effect on the global changes in K4 monomethyl dimethyl and trimethyl. Now, we've never gone and done a ChIP-seq to look if there's specific gene loci that have been lost or changed over time, but I think it's pretty clear that there's no major effects that was as predicted in these mice. So if you look for what SET7 methylates, it's a great methyltransferase. It can methylate just about anything. And so from P53 to important things like some TAFs, or PRB, STAT3, P65, all of these. I think we're up to 27 right now. 28 is at the last count. Well, I guess 29 with the one that we found now. And really, all of this has been shown that if you take SET7 purified and you mix it with a protein, it will methylate it. And it's pretty clear that happens to a lot of these things. And so a lot of these studies have been done using that, and very few of them, in fact, none of these were done looking in vivo at the effects, because we know that genes like P65, TAF10, P53, they have dramatic effects on disease development, on survival, on development. MECP2 mice are embryonic lethal, DNMT1 mice are embryonic lethal. All of these sort of things that we thought, if SET7 was really having a dramatic effect on them, we should see some effects in our SET7 knockout mice, which we don't see any difference in development, and we see no effects really on cancer progression, et cetera, in the steady state. And so we really wanted to start thinking about, well, what's going on? And so a postdoc came to the lab from the Netherlands, Menno Odoff, and he was an epithelial cell biologist. He worked on wound healing in epithelial tissues. And he wanted to really look at epithelial cells. And so the way that we addressed looking at SET7 was he made a SET7, using SET7 floxed mouse that was generated by Fabio Rossi at UBC, crossed to the Villain-Cree. And so we had specific deletion of SET7 in the IECs, and we looked for a phenotype. And the first little while was there was very little phenotype observed, but because he was really sort of quite anal and looking at things, this was the phenotype that he found that led to all of the studies. Was this? This is the control mouse here. This is large intestinal crypt. You can see this is sort of looking normal, and this is the knockout crypt. You see it's a little bit wider and it's a little bit shorter. And when you stain it for EDU, staining for proliferating, we see a few more proliferating cells per each crypt. And so this phenotype was really reminiscent of a phenotype that was observed in another strain of mice that was related to the Hippo pathway. And the Hippo pathway is a pathway that's been recently shown to be involved in cell proliferation, cell survival, and tumor growth as well. And I'll talk a little bit about more details in a second. But why did we have larger and wider crypts? And so there was a couple of possibilities here on the interaction between this methyltransferase and the pathways involved. And so what we found is, briefly so in the stem cell compartment, you have these intestinal stem cells that sit between Paneth cells at the base of the crypt that can be proliferating. You get transit amplifying, you get proliferation, expansion, and then you get differentiation as they go up there. And there's a few pathways that are associated with all of these different things. It's not perfect, it's not exactly black and white, but this beta-catenin is really important in this stem cell pathway. Hippo has been shown to involved in proliferation and cell size, and Notch is really important for differentiation, either promoting or inhibiting the development of different intestinal cell types. So what is the Hippo pathway? So the Hippo pathway was first described in flies that if you mutated parts of this pathway, tissue overgrowth. So really, imaginal wing discs would grow 5, 6, 7 times in size. So it really was one of the factors that was thought to really control, well, why do our lungs grow to a certain size? Why does our liver grow to a certain size and fit in our body? Well, this is really one of the pathways that controls that. And so really one of the major first things that was described was cell-cell contact. So as cells grow in a dish, the Hippo pathway is inactive. And when I say inactive, that means that there's no signaling through this MST1 and LATS1 pathways to phosphorylate the transducer of this pathway called YAP. So YAP is remaining in the nucleus. It's transactivating genes that are associated with cell growth, pro-survival genes. It interacts with this coactivator is a coactivator for this transcription factor called TEAD. It's constantly cycling back and forth into the cytoplasm. And if the pathway does get activated, so at high cell densities, when cells start touching each other, there's a kinase cascade that results in the phosphorylation of YAP, which leads to multiple phosphorylation, ubiquitination, and then degradation. Or, in contrast, it can also be bound by other factors and be sequestered into these junctional places. So the activation of the Hippo pathway leads to turning off of YAP, and so the loss of YAP activation. And so through a bunch of stuff I'm not going to get into, we focused in on this pathway when we were looking at our phenotype with the SET7s. And really, this was Menno's paper from 2013, where he was looking at the interactions between SET7 and YAP. And what we found was that SET7 and YAP interact. SET7 methylates YAP on a specific lysine. And really, we can identify, the whole paper can be summarized, about 3 years worth of work can be summarized in this one figure. In that, in a steady state, here's a sparse cell population. Now, these are transfected cells, so they're overexpressing it. But you can see that the nucleus has YAP in it at sparse. If you let those cells go dense, you can see a lot more nuclei there. You can see that the nucleus, YAP gets excluded from the nucleus. We're overexpressing, so it's not black and white. We've got some pictures of that. But if you mutate a single lysine in the YAP tail, this is at the very end of YAP. YAP's about a 502 amino acid protein. So there's a lysine in the C terminus of it. Then the YAP doesn't leave the nucleus even at really high density. If you mutate a lysine just downstream for that, then it leaves normally. So we use this to sort of suggest that SET7 can methylate YAP, and this is really important for its subcellular localization. Now, we have no idea how this works, what the methylation is doing. We've got lots of negative data to show that it doesn't affect proteasomal degradation, doesn't affect sequestration, how that is controlling it. Obviously, it's creating a binding site for something else to bind to it, but we don't know what that is yet, and so that's still going on. But in the process of doing this, it sort of led us to a couple of, this is the subs, yeah. So we've got SET7 here, and data that I didn't show you is that turns out in our hands, SET7 is both cytoplasmic and nuclear. And in fact, this data right here could be set7 as well. At low density cells, set7 is in the nucleus, and at high density, set7 is in the cytoplasm, sort of mimicking what we see with YAP. So we've got this protein, this methyltransferase, that seems to be modulating its localization as well, depending on context, cell-cell contact. It can interact with this protein YAP, and it can methylate it. And this methylation event is really critically required for it to be maintained in the cytoplasm at high density. So, in the context of while we were doing these experiments, and this is just, I guess, where serendipity comes into, some collaborators at the Structural Genomics Consortium. This is a public-private partnership between a bunch of different companies, about 7 or 8 different companies, including Pfizer, and the University of Toronto and Oxford University, where they have a mandate to generate chemical probes, inhibitors against all the epigenetic modifiers that they can find, from acetylases, methyltransferases, deacetylases, demethylases, BRD binding things. This is where the JQ1 BRD inhibitor that a lot of people might have heard of in cancer treatment came from, was through this interaction. And people at Pfizer, John Tatlock, who is the chemist behind it, and David Owen, who sort of leads the interaction between them, developed this drug called rPFI2. And it inhibits set7 activity in vitro really well, so down in the submicromolar concentrations. What's really great about it is that it has an enantiomer that is less active. So we have a less active enantiomer. So when we use lower concentrations, we have a great control for both of these things. And we published this paper a couple years ago showing that this drug actually inhibits the binding of S-adenomethionine to the pocket, has a really novel mechanism of action, but it mimics what we see in the set7 knockouts, in that if we take the active drug and we look at high density, you can see, here's the inactive, this would be like the wild type controls. At high density, you can see that YAP is found less in the nuclei and found sort of at these junctional things. If we add the drug, we can keep the YAP in the nucleus at this high density here. And this is just, quantifying all of that. So what this suggests is that the effects of set7 deficiency were mediated through likely mediated through its methyltransferase domain. So this sort of, again, suggested that set7 was methylating YAP. And this methylation event was important for its subcellular localization, and it had an effect on gene expression. These are some of the canonical genes that were increased when we treated them with the drug. So with that sort of background, this is where we sort of got to, and we're thinking about, well, what's going on in the gut? Why do these mice have shorter crypts, but wider crypts, and what's the cells that are involved in all of this? And we started thinking about these other pathways that were associated with all this. And during this time, a whole bunch of papers came out in single-word journals telling you that the Hippo pathway and the Wnt beta-catenin pathway were closely related. They all said something different, and they all said controversial things and contradictory things, but it was clear that there's something going on. There was some link between the Hippo pathway and the Wnt signaling pathway, and this was in heart, this was in tumors, and this was also just in the steady state. So we started to think about, well, what is the interaction between YAP, and does the methylation of YAP and set7-dependent effects have any effect on Wnt beta-catenin pathway? And this is where, you follow biology and it takes you in places you don't want to go, right? I'm not a T-cell biologist, I'm not a tumor cell biologist, and I'm not a Wnt signaling person too. So this took us a while to sort of get my head around, but really Wnt pathway signaling is really the most important part of it is the beta-catenin protein, which is, again, like YAP, regulated very similarly, except for in the opposite direction. It's kept in the cytoplasm in this so-called destruction complex, which is a complex of several proteins that lead to its phosphorylation and degradation in the steady state when there's no Wnt. In the context of a Wnt signal, something happens. Again, this seems to be pretty controversial, how this is regulated, but it leads to the accumulation of beta-catenin in the cytoplasm, migration of beta-catenin into the nucleus, where it replaces these repressive complexes, those Groucho TCF complexes, to become an activating complex and leads to gene expression and proliferation and expansion. And so we wanted to start thinking about, well, how does the set7 pathway impinge upon Wnt beta-catenin if YAP and beta-catenin are interacting in some sense? And so what we started to do is just take our drugs that we had. This is using 293 T cells stimulated with Wnt3a for a variety of hours. And what you can see is that this is, again, these are cytosolic Western blots. So you can see there's low levels in both cases. And if we stimulate with Wnt3a, we can see this rapid accumulation of beta-catenin in the cytoplasm. SET7, again, doesn't change and all that. So there's no effect on the activation of this pathway. So blocking SET7 or not blocking SET7 really has no effect on the accumulation of beta-catenin. But what we did see a striking finding was that when we stimulate with Wnt, we see a nice increase in axin 2, which is the canonical readout for Wnt signaling. If we do this in the presence of the inhibitor of SET7, we can block that activation completely. And so we lose this. So blocking SET7 blocks Wnt-dependent, beta-catenin-dependent gene expression in response to Wnt signaling. And because we saw this huge change in YAP localization in the context of SET7, we first looked at nuclear localization of beta-catenin, and this is where we see a really strong phenotype. Whereas we can see nuclear accumulation within 15 minutes after stimulation with Wnt in the context of the negative inhibitor, of the negative control. What you can see is that we fail to see beta-catenin getting into the nucleus in the absence of SET7 methyltransferase activity. So somehow SET7 is interacting with this pathway to limit its ability to activate this pathway fully by driving beta-catenin into the nucleus. So SET7 does interact with beta-catenin, and so we can show that. So if we do an IP now for SET7, these are all native IPs, you can see that beta-catenin can get pulled down with SET7 in the presence or absence of the methyltransferase activity. So SET7 binds to beta-catenin even if its methyltransferase activity is being blocked. But what's interesting is that when we add Wnt, this interaction is gone. But if we add Wnt in the context of the of the inhibitor, that is maintained. So this suggests that there is SET7 methyltransferase activity has to be intact during Wnt activation for beta-catenin to be dissociated from this part of the complex. Now, in converse, what we see is when we look at the YAP and beta-catenin interactions, like it has been published before, that we had to do this, I think we did this in a overexpression, I can't remember now, actually I can't remember. But you can see it's a lot less. When we IP YAP, we can pull down a little bit of beta-catenin. If we activate the Wnt pathway, we can see maintain this. But if we activate the Wnt pathway in the absence of SET7 interaction, we lose this, we don't get this interaction, although it's faint there. So that suggests that the interaction, so SET7 interacting with the YAP and beta-catenin pathway suggests that there's a complex of YAP, beta-catenin, and SET7, perhaps other things as well. We haven't really delved into that really deeply biochemically. That when the Wnt pathway gets activated, SET7 leaves that complex. But the methyltransferase activity of SET7 is required for YAP to be maintained a part of that complex. And if the YAP is lost from that complex, then beta-catenin doesn't enter the nucleus. So this sort of suggested that YAP and SET7 were interacting. The methylation of YAP was really critical for the complex, for this complex to work, for beta-catenin to get into the nucleus. And this is a really busy slide to show that this is actually dependent upon YAP. So let me just see if the lines are on. No, I didn't. So in this slide, what you see is if you just focus on the axin 2, you can see these are using siRNA now to knock down various components. You can see that if we knock down SET7, we lose the Wnt-dependent axin expression. But if we knock down YAP, in fact, it goes a little bit higher, actually, when we lose the YAP. But when we knock down YAP and SET7 together, we don't have this reduction. So losing SET7, the loss of SET7 requires YAP to control that differentiation. So SET7 and YAP seem to be really important for driving Wnt signaling in these cells. So we next wanted to ask, well, as I said, the SET7 mice have no phenotype whatsoever. There's no cancer development in the steady state. There's no, they live normal lives. Their guts look absolutely fine. They have the same number of Paneth cells. All these things that we think of as Wnt-dependent processes. You know, in our cells, when we inhibit this thing, we get really poor Wnt activation, yet our mice develop normally and develop fine. And so we started thinking about, well, what's the difference? And so the big difference is that we're giving Wnt's, really high levels of Wnt's to these cells, and we're seeing an effect. And so there are some cases where high Wnt signaling is different from low Wnt signaling. And so in processes where you need repair, like intestinal regeneration, or in tumorigenesis where there's a real strong Wnt signal, we thought that maybe there'd be an effect of SET7. And sure enough, that's where we started seeing some major effects. And so this is the model that we first used. This is a model of intestinal regeneration following 15 gray irradiation. So you irradiate the mice, a whole body irradiation, and their guts get pretty destroyed. They get pretty sick. So this is 3 days after. And you can see you lose all the crypts. This is the small intestine. They look pretty terrible. But by day 6, wild-type mice start to repair. You can start to see some crypt fission. You can see these normal crypts coming back. We never see that in the absence of SET7. So in these guys, we're seeing really poor development of crypt architecture, and there's very little recovery that we've quantified over there. So in this context, SET7 is absolutely required for the development of a Wnt-dependent response. The opposite thing is what happens if you have too much Wnt. And so this is a model of tumorigenesis. So the APC min mice that have a mutation in the APC, part of the destruction complex, constant beta-catenin activation, constantly having beta-catenin in the nucleus. We've crossed these to the het so that we can see that there's a delay in the time to disease. So we have a significant delay in the time to disease. They still get cancers, and they still get sick, and they do die, but the tumors that develop are significantly smaller. And if we just look at this is the small intestine, this is the large intestine, they get a lot fewer tumors. We see very, very few tumors. If we take those same mice and we put them on DSS, induced intestinal inflammation, you can induce tumor burden in the large intestine a lot quicker. Even those are a lot smaller and a lot less well-formed. Now, our data was suggesting that the major defect was the movement of beta-catenin into the nucleus. And when we started looking at the tumors that were developing in these animals, what we found was pretty striking. So this is the wild-type tumor, or the het tumor. You can see this is staining for beta-catenin. You can see it's throughout the whole cell. And I don't know if you can see it on here, but in the tumor of the knockout mouse, you can see these white spots all over, which are the nuclei of these cells. So we are still seeing a lack of beta-catenin in the nucleus of these cells. So we're suggesting that maybe these tumors are different, are distinct, and could be mediated by other effects, but we still haven't followed that up yet. But it's clear that there's a link between SET7 methylation of YAP leading to the control of the beta-catenin localization, somehow linking it to tumorigenesis. So we published this just a couple months ago, linking SET7, somehow linking these two pathways together. And we think that YAP interactions with this destruction complex or with this is really dependent upon this methylation event that's mediated by SET7. Now, there's still lots and lots of questions. It suggests that SET7 might be an okay drug for maybe treating tumors, but you also get irradiation, and it's probably not a good drug to be using if you're going to be having any kind of intestinal damage or regeneration. And so it has a lot of interesting questions, clinical questions, and also some interesting effects on whether this is actually going to be an important pathway. It also suggests that these pathways are maybe not two separate pathways, but maybe are considered one super pathway and are really regulated. They're regulated by the same sort of proteins, the same proteins that methylate, phosphorylate beta-catenin, also phosphorylate YAP. And so it seems like there could be a super complex here that's involved in controlling all of these aspects of this pathway. So I'm going to switch gears, and then I'm going to move away from the tumor cell stuff and the methylation of non-histone proteins to move towards sort of the canonical forms of methylation, at least in the system, and that's controlling gene expression in the context of histone modifications. And the one I'm going to talk about here is this one, G9A, which again is the histone methyltransferase. Its main role is as a histone dimethyltransferase. It's pretty striking that if you knock out G9A, you lose specifically the dimethylation. You don't lose the trimethylation, and you don't lose the monomethylation, which is it's a pretty specific effect. G9A forms a heterodimer with this partner G9A-like protein, or GLP. And if you mutate G9A or GLP, you lose the mechanism. So these they're absolutely required to both these partners to be there. There's some thought that maybe they give different results if you have different knockouts, but it's still not that clear. But in general, we're using this one, so we lose the GLP does not compensate for the presence or the loss of G9A. So G9A is a large protein. It's about 1,300 amino acids, and it's made of these domains. There's an N-terminal domain that was, up until recently, not really known what it did. It has a series of these ankyrin-like repeats, and these ankyrin repeats were shown several years ago that they form a cage that then can bind to dimethylated lysines. So G9A can dimethylate H3K9, and then it can actually bind to the mark that it lays. And it's thought this is one of the mechanisms of how perhaps epigenetic marks on histones can be translated across several nucleosomes. And so the idea was that it would bind it, it would lay down this mark, it would bind to it, and then it could move to the next one in the context of that. It doesn't have its own DNA binding domain, so it has to interact with other proteins in order to get to the DNA, in order to mediate these effects. I don't think I showed, yeah, I didn't. So this N-terminus here was just recently shown by Mike Stallcup, who's at University of Southern California, that in some contexts, G9A can be an activator. It's really well known as an inhibitor, as I'll talk about, but this part of the protein can interact with other transcriptional co-activators to mediate transcriptional activation, and I think I'll mention a little bit of some of our data on that. But in general, Yuichi Shinkai made these mice about 15 years ago now. The total knockout mice are very early embryonic lethal. And a lot of the work that's been done on this pathway in these mice has been done on embryonic stem cells. And what's really interesting is that the embryonic stem cells that they get out of these mice, even at this early time point, express a lot of genes they're not supposed to. They express a lot of spermatogenesis genes. They express a lot of other proteins they're not supposed to be expressing. And you can take those ES cells, differentiate them to neuronal, to fibroblast pathways. If you remove the factors that drive that, they will de-differentiate back into ES cells. And so this was really some of the data suggesting that this is a mark that's really required to repress gene expression, specifically of pluripotency genes, so shut those down to allow cells to translate and become a differentiated cell. And in fact, in some of the early iPS cell papers, simply including an inhibitor against G9A could replace some of the factors to generate iPS cells. So just blocking G9A would allow you to de-differentiate cells in the context of a few less factors. So in general, it's been thought from ES cell studies that G9A and its mark, K9 dimethyl, was a repressive modification that was required as cells differentiated to shut down promiscuous gene expression and maintain lineage integrity. And so as a T-cell person, this was ideal. This was perfect because we thought we can look at this in the context of T-cell differentiation, which is exactly what we expected. As T-cells become from naive, they become Th1 or Th2. They don't express the other genes of the other things at high levels. And so perhaps there was an epigenetic mechanism, and this would be the mechanism that we found. And of course, we were wrong. But just some background about this. And so really, this is sort of the histone code, as has been called, is that genes that are expressed tend to have modifications associated. And perhaps the most common is the MLL-dependent K4 trimethylation. And the inactive genes tend to have marks this EZH2-dependent K27 trimethylation, but G9A is also really important in this sort of inactivation. And again, these are associated with expression. They're not absolutely required, and I think there's enough data out there suggesting that you don't need this mark to get gene expression, but it's associated with it. And so how that is thought of is changing as we go along. Now, there are populations of genes that have both modifications on them. And in embryonic stem cells, this is really clear. There's a lot of these genes that are rapidly activated or repressed based on having both of these modifications. Just the removal of one of them was enough to lead to turning on or turning off of a gene and through that. And there's a whole set of these. And Steve Turner, who we've now poached over at Monash, has a nice study in CD8 T cells showing that there's a whole bunch of these poised genes that are sitting there in naive T cells, and then compared to the memory cells, which ones go on and go off. There's a real nice set of genes that are associated with this poised phenotype. And so we thought this is the perfect place to start looking at epigenetic modifications, because in Th1 cells that respond to IL-12, they express transcription factors like STAT4 TBET, they make interferon gamma, whereas like Th2 cells express STAT6 GATA3, express IL-4, 5, 13. these guys tend not to express this, and these guys tend not to express this. So there's a mutual exclusivity in their expression profiles, which likely is going to be controlled, at least at some level, by some of these epigenetic mechanisms, or there should be some explanation, some interaction between the two. I don't think that. Yeah. So around this time, Keiji Zhao, who's at the NIH, did a big ChIP-seq study on this, and showed pretty well what everybody expected. It's not too dry. Is that naive cells have very few marks, or they have a couple that have this poised phenotype. As you make a Th1 cell, genes that are expressed have an activating mark, genes that aren't expressed have an inhibitory mark, and this is true for most things. But there were a few surprises in that some of these populations actually maintained this poised mark. So regulatory T cells that don't express T-bet GATA3 or ORC maintain this poised phenotype, and this perhaps suggests that plasticity is maybe more of the rule, and this happens again in Th17 cells and at some aspects of these Th1 and Th2 cells. So there's these alternate opposite transcription factors that are maintained in this state that could be activated at a later time. And so it's not really clear how this is, but it really suggests that there are multiple layers of mechanisms that were controlling this, these pathways and these lineages or these subsets of cells as they were developing. So I'll summarize a little bit of what we did with the T cells, because I'm going to spend the last little bit about some of the work that the more recent paper that we've just published, and that we found that in the absence of G9A, in a T cell intrinsic manner, G9A is required to promote T cell differentiation. And we have a really strong defective T cell response in the absence of G9A, and we think this is due to that first 300 amino acids in the protein, because it's independent of its methyltransferase activity, this promoting effect. And so there is a role for G9A in promoting gene expression, and we think it's somehow interacting with this. The same data has been found for the beta-globin gene cluster. So G9A is an activator of the beta-globin gene cluster, and the beta-globin gene cluster is almost identical in its sort of gene structure to the IL-4, 5, 13 locus with the LCRs and the gene arrangement. So we think there's some genetic interaction, but maybe not between these two pathways, but between the way that these genes are set up, that G9A acts as an activator in there. I'm not going to talk about any of that. We still have no idea about that. But one of the things that we found in this early paper was that we found IL-17 production under Th2 conditions in the absence of G9A. And we sort of just mentioned it. We didn't really follow it up until a few years later, when we found that actually G9A is really a critical modification that limits Th17 and Treg cell differentiation. And so in the absence of G9A, or in this case, in the presence of its inhibitor, so this is a strong epigenetic regulation pathway, we see high levels of Th17 and Treg cell responses, both in vitro and in vivo. And so we think that G9A has these multiple roles in T cells that leads to regulating the cell lineage fate, although it's very different from what we see in embryonic stem cells. But what I'm going to spend the time on is that we took that information that we had from the T cells and we thought, well, what about innate lymphoid cells? And of course, it wasn't that simple. What we had done is we had done an assay where we had thought, well, we have a defective Th2 response. Let's look at a disease like asthma. And so we did the we did an asthma challenge. We did a couple models of it. And what we found was that there was a slight defect in the development of disease. There was less disease. There was a little bit less of a Th2 response, but it wasn't completely gone. It wasn't until we knocked out G9A in all the hematopoietic cells when we could completely abrogate the development of asthma. So it suggested there was something else going on. And our first target was actually back 5 years ago was mast cells, but in the context of all the expansion of this population of cells, these innate lymphoid cells, we started thinking about the role for G9A in other lymphoid cells, like the innate lymphoid cells. And the ILCs have really expanded since their sort of, I guess, their discovery in 2010, I won't say, or just description, I guess, in 2010. They've really exploded onto the scene as a population of cells that seems to do everything that we couldn't explain before. And I think this sort of fits into a lot of pathways that people were sort of hand-waving about. Now these cells seem to be popping in and explaining a lot of the differences that we were observing. And just like T cells, these can be grouped into different subsets. These are CD3 negative lymphoid cells that can be grouped into ILC2s, which are akin to the Th2 cells, which I'll be focusing a little bit on today, as well as the ILC3s, which can then be subgrouped into different subsets of these, sort of like the Th17 cells. development of these cells, again, is coming from this common lymphoid precursor through a whole range of this, and there's lots of studies now that have been showing the cells that drive and are required for the development, the factors and the pathways that drive the development of ILC2s and ILC3s from these progenitors that are found in the bone marrow. And so we started thinking about the context of ILC2 activation in G9A because we were seeing this defect in Th2 cell responses, so we wanted to address whether the ILC2s are being activated or dysregulated in any way as well. And a lot of the work in the ILC field has been focused on the ILC2s because quite a bit is known, is that they respond to a wide range of cytokines. Most of these cytokines come from epithelial cells or damaged epithelial cells, so IL-25, TSLP, IL-33, that act on these things to induce high levels of cytokines like IL-5 and IL-13. And this seems to be really critical for early activation of immune responses at barrier sites. And so the first thing we did is we started looking at whether the absence of G9A had any effect on the development of ILC2s. And what you can see is now this is a immature ILC2s. This is a bone marrow stain. We're looking at lineage negative cells. We're looking at c-KIT SCA1 and c-KIT low, SCA1 high cells. And of those, we see a nice population of CD127, CD25 positive immature ILC2s, that's the mark that we've used. And what we could find is that we knock out G9A using a VAV-CRE, which knocks out in all hematopoietic cells. We can see a severe reduction in the frequency of those. We see an increase in a sort of a block almost back at this stage here, and the cells are sitting in this negative spot, and they're not getting into this immature ILC2 phenotype. Whereas we knock it out in the T cells, again, we see normal numbers. When we look at those cells, the wild-type cells express normal levels of the 33 receptor, ST2, whereas the knockouts, we have about 50% of the cells that can actually express it properly and 50% that don't. So we have this sort of this biphasic expression of ST2, which again results in a strong defect in the number of immature ILC2s that we find in the bone marrow. So I think this was an experiment the reviewer asked for, but so we did some bone marrow chimeric experiments, and what we showed is that even to get a 50-50 in bone marrow chimerism, we had to put in about 5 times as many G9A knockout bone marrow cells as we had to for the wild types. But once we got to a 50-50, sorry, there's the 50-50. So once we got to a 50-50 with a non-ILC, what we could find is that we could not get any knockout ILC2s from those bone marrow chimeras. So there was a real striking difference in the ability of these cells to develop and maintain an ILC2 phenotype in the absence of G9A. When we look now at more mature ILC2s are found in a lot of tissue sites. You can find them in the mesenteric lymph node, but you can find them in the lung and in the visceral adipose tissue at sites. And everywhere we looked, we always saw this strong reduction of them. We could see some. There was always some there. We could always find a few, but they were always a significant reduction in the numbers of those ILC2s that were found in the tissues. And when we look closely at those, and we look at the numbers, what we could find is that the ones that were in the tissues, in these expressed normal levels of the receptor ST2, as well as IL-25 receptor. So the ones that could get out and get to the tissues seem to be fine. They seem to express normal levels of the activating cytokines that we would have expected, although there's a lot fewer of them there. However, when we look at them on a single cell level, so what we did is we went and sorted out ILC2s from the lungs. We could only get very few numbers of cells, but we would put them in there, and they make so much cytokine. It's actually easy to measure the cytokine coming out of them. We activate them with cytokines, and what you could find is even in the context of equivalent cell numbers now, we still had a reduction in the activation of those cells. They just would not produce same levels of IL-5 and IL-13. So there's a developmental defect in these cells, that there's a lot fewer of them, but they also seem to be on a per cell basis less functional. Now we have no idea what this means. We have no data on whether this is a 33-dependent effect, a TSLP-dependent effect. We've tried this with adding an IL-25 and taking out IL-25 and all the different combinations, and they always are reduced. And so we're trying to figure out exactly what that is, but we don't have the answer to that yet. So this is all done, those experiments are all done sort of in vitro, so we wanted to ask if this defect in G9A and the defect in ILC2s had any effect on disease burden and development. And so we treated these animals with papain intranasally, and this is a model where you get damage to the epithelial cells, you get release of these mediators, and you get a nice ILC2 induction, and it's very rapid. So within 3 days, we can get a nice lung inflammation response that we can enumerate. And so what we found is that when we do this into the wild type or into the VAV G9A mice, we could see a reduction in the bowel count. So again, not gone completely, but reduced. But what was striking was the biggest effect was on the eosinophil influx. And so we saw no difference in the macrophages, the lymphocytes, or neutrophils in a bowel differential, but we could see a striking effect on the eosinophils, which was associated with the loss of less goblet cell hyperplasia and inflammation in the lungs. So again, these mice seem to be protected from the eosinophilia associated with papain, which is definitely an IL-5 mediated event, which has been associated strongly with defective ILC2s. So we wanted to ask, well, why are there no ILC2s in the context of this infection? And so what we did is we took our immature bone marrow cells, ILC2s, we grew them in vitro for 14 days in the presence of IL-2, 7, 25, and 33. We grow them out, and then we just did an RNA-seq. And what we found was pretty striking, and it's pretty clear, is that our ILC2s were becoming ILC3s. And so you can see that the genes that are upregulated, I just picked out a few of them here that are associated with ILC3 differentiation and development, so IL-17, RRC, IL-23 receptor, this is the ST2. As well as the genes that have been associated more with, this is the ST2, with ILC2 differentiation are downregulated. And so it's pretty black and white, actually. So we're seeing a switch in the phenotype from an ILC2 to an ILC3 phenotype. And when there's been a couple of phenotyping consortia that have been sequencing canonical and coming up with canonical gene lists for these different subsets. And so if you take this sort of 100 ILC2 core genes and you just rank them, you can see that the wild types express most of those at higher levels than the knockouts, whereas the knockouts express only a few of them dysregulated sort of unpredictably. And the ILC3 core genes are exactly the opposite. The knockouts express a whole range of them. And these are genes that you never would have predicted to be up, but of course, because they're part of this ILC3 core phenotype, they are upregulated. So there's this dramatic increase in the switch between ILC2 and ILC3s. Now, this has been shown before for other proteins. So GFI1 is a growth factor independent one, has been shown by Harinder Singh to be required for this to control this ILC2 and ILC3 switch. And in those mice, in the absence of GFI1, ILC2s start making ILC3 cytokines like IL-17 and IL-22, and they start producing these things. And it's pretty striking is that one of the great binding partners for G9A is GFI1. So they interact with each other, and it's been shown in multiple cell types that G9A and GFI1 are binding partners. So we thought this is what's happening. Our cells, through GFI1 interactions, are now becoming ILC3s. Except it's not that simple because our cells are not becoming ILC3. So what we found is that none of our cells are becoming double producers. So this is the wild types ILC2s. We see lots of IL-5 production, very little IL-17, A or F. Whereas in the absence of G9A, what we find is that we see a reduction in the number of IL-5 producing cells, but we see a huge increase. We see an increase in the frequency of IL-17 producing cells in the absence of G9A. And these cells are never producing from IL-5. So what we think we have is we have a population in the bone marrow that's growing out. And I think it's the ST2 negative cells, that 50% of those cells that are growing out, that those cells can then become ILC3s, and I didn't put the data in here, I didn't think I'd have time, but that's what we did. When we sort out the bone marrow, the ST2 positive and the ST2 negative cells, and then we do a transplant into non-skid mice, common gamma chain knockout mice, what we find is that the ILC2s that are ST2 negative give rise to ILC3s, whereas the ILC2s that have ST2 give rise to ILC2s. So we think that there's an early event that's mediated by G9A in some way that's required for the optimal expression of ST2, which leads to the development of this of these cells, and that in the absence of G9A, we're allowing the development of IL-17. So I won't show the data, but this is definitely dependent upon its methyltransferase activity. So the activating form of G9A doesn't seem to be playing a specific role in this, because if we use an inhibitor, we can recapitulate all of these events just by using a G9A specific inhibitor. And so the question was, well, is this happening through its methyltransferase activity? Is this happening through its epigenetic mark? And I don't think we have the exact answer to that, but it's clear that there is an association with the two things. And so we can see that in the absence of G9A, so K9-dimethyl, again, it's in general, a repressive mark. And so in ILC2s, these are in grown out ILC2s out of the bone marrow, what you find is that we see very low levels of IL-13 at the IL-13 and IL-5 promoters. And this is because these genes tend to be expressed, so we see very low levels of that. And what we find is that we see high levels at the IL-17F promoter. And then in the absence of G9A, as you'd expect, we have low levels of K9-dimethyl across these things. So what we think is happening is that the lack of this mark at these loci is allowing genes that are normally repressed to be transcribed, and that's leading to the differentiation of these cells into the wrong phenotype. So, summarize that part is this: G9A we think is really playing a really important role in lymphoid cell biology, and really it seems to have a real major effect in controlling these ILC3/Th17 gene programs. And I didn't show you any of the data in the Th17 cells, but it's really clear that in the absence of G9A, you get a much higher Th17 response, although you do get much higher Treg response. So there's some linking to this, and evolutionarily why G9A has been co-opted to control this gene program. It has nothing to do with G9A dimethylation of K9 has nothing to do with Th1 and Th2 cells, because there's no effect on any of those in repressing those marks. And so why it's been co-opted to repress this pathway and not the others is really kind of an interesting thing, an interesting thought that we're still trying to sort of put together, and sort of identifies G9A as this central way of maybe perhaps, in context of, wanting to modulate or improve this, could be an interesting target. So this actually is not them, but yeah. So this work was all done by, the G9A work was all done by Fran Antinano, who's a postdoc in the lab, who's still in Vancouver. Menno has his own group in Norway. He's the cool dude, yeah. And Sebastian he's the favorite one because he came with me to Melbourne. So he's here in Melbourne now. So he's at Monash. And these are my two students. Alistair just graduated and is looking for a job, and Kyle will be graduating, but Kyle's working on the Hippo work. And so again, a lot of this work was in collaboration with Fabio Rossi and Bernie Lennertz. And the people at the SGC in Toronto have been outstanding with their help. And the Set7 work with David and John has been great. So. Thanks so much for listening, and I'll take any questions.
Thanks, Colby. That was great. Any questions, particularly students? Yep.
Do you think that such main proteins methylate in a stepwise manner? And if so, does dimethylation have a real functional consequence? Or is any effect you see upon loss of dimethylation really because dimethylation then can't occur?
So that's a good point. So, and I didn't show a lot, and I glossed over a lot. So in the absence of G9A, you don't have any K9-dimethyl, but you have normal levels of trimethyl. Okay. So it's not, in some cases it is, in some cases it's not, I think. I think that, and it all depends, I think, on, it's all about steric hindrance in the pockets of the binding. So Set7 is a monomethyltransferase. It cannot do dimethyltransferase. They've tried fitting dimethyl, so it can't dimethylate because it just can't structurally accommodate it. So I don't think so. But I think what's interesting thing is that there are proteins that will specifically bind mono, di, or tri. So I think having all three of those does give different possibilities. And so that's why I think it's even almost more dynamic than what phosphorylation provides, because you can have a monomethylation event that is different. So monomethylated K4 and dimethylated K4 is found at different spots than trimethylated K4. And so why that is and how that's controlled, I don't think anybody has any idea. But yeah, that's a good point.
Yep.
So earlier on when you showed the diagram of the crypt and the creative difference in pathways not showing the Hippo. So the Hippo, do you reckon the signal that occurs in the transient amplifying sort of have a gradient?
So that's a great point. So I put that up, that's kind of just really, so I think it sort of goes through all of that. But if you look at where YAP, so if you do a stain for YAP, it's actually nuclear in the bottom, and it's cytoplasmic at the top. And as it goes up, it becomes more cytoplasmic across the crypt. So I think, and the same thing for beta-catenin as well. It changes as they go up. So I think there's going to be, so the cell contact has a big effect. There's also GPCR signaling. There's a bunch of other things that are now leading into Hippo and Wnt signaling as they go up the crypt. So yeah, absolutely. So if you just do a stain, you can see it sort of moving out of the nucleus. And so the endocrine cells at the top of the epithelial cells at the top, YAP is all cytoplasmic. And in the base, it's all nuclear. And so what's the difference between that? Yeah, I don't know what the signaling is that leads to that, but yeah.
Hi, Eliza. Can I ask you two questions?
Yeah.
So one is about the tissue residence characteristic of the LC2. I was wondering if perhaps if you have this lower number in the lungs, if it's because the deletion of gene 9A is affecting their homing into the tissues. Have you tried to do, like, inject IV anti-CD45 antibodies to see if they are in the tumor, in the lung parenchyma or closer to the
Haven't done that at all. When we transplant, yeah, so how have we done that? Have we addressed that? No. So absolutely it could be that. We've looked in the blood, though, and they're lower in the blood as well. So there are not very many in the blood. The numbers are so low. I can't say it doesn't have, it's not the question, not the answer, but I don't know. Yeah.
No problem.
Yeah.
And the other question is, what's the phenotype on the VAF3 G9A on ILC1s and NK cells?
So ILC1s don't know, NK cells, so we haven't looked at that. The ILC3s, as you'd expect, they have about 5 times many more ILC3s. But that's maybe because we see that. So that's it. And they're much more resistant to Citrobacter. So we can give them a lot higher dose of Citrobacter and they'll become clear Citrobacter. But for ILC1s and NK cells, no idea. You can do that if you want. So.
If there's no more questions, I'll get you to join me to thank Colby. And there's also lunch for the students, I think, in the boardroom.